Hello children, we are going to take class 12 biology, chapter 5 principles of inheritance and variation, part 1. This is the unit of genetics and evolution. Do you know why the elephant give birth to an elephant only? Why a mango tree gave only a mango fruit and not any other else? What is that? It is making or it is carrying the information from the parent to offspring. Can you imagine or have you seen the parents exactly similar to their offsprings without a single difference? Or do the siblings they have very similar characters? Are they exactly similar? No, they are not exactly. There is a small variation is seen. So scientifically, we study this under the branch of biology called as genetics. And this subject deals with inheritance as well as variation, which is transferred from parent to offspring. What is inheritance? It's a process by which characters are passed from parent to progeny. So we carry our character or we get our character from our parents. And this is the basis of heredity. Now, how variation is formed? So there is a variation in each and every individual. That's why the fingerprints are different, the color of eyes are different, the blood groups are different in a same family or in a same parental offsprings. This is a cow which is a which, which is a Shahiwal, and it is produced through hybridization technique. So we can say that the process of genetics was earlier known to, and our ancients they performed several experiments and they produced varieties of fruits and vegetables of a same species. We have large number of mango varieties, we have large number of rice varieties and etc. So the science was known earlier, but now it has moved to its highest, greatest step. So the chapter is mainly based on Mendel's laws of inheritance. So the Mendel's started his laws or stated his laws with the help of experiments that he was going to perform. And the experiment was called as the hybridization experiments. He used garden peas for 7 years and proposed this law. And he used his mathematical logic and problems, and he collected statistical data and analyzed them to come to the confirmation and stated his rules or the laws. And his experiments they were of large sampling size and they were of greater credibility because of the large sampling size. He used pea plant. Why? He used pea plant because pea plants they show various contrasting traits. I call them as characters, traits or characters. But he used only 7 of the characters for his experiments. He used pea plant also because it was easy to grow and it showed several contrasting traits, and in a single season more than 2 generation of this pea plant can be obtained. So he used the 7 contrasting traits for his experiments. The flower color purple or white, the plant height, the seed color round wrinkle, pod color the green or yellow, the pod shape inflated or full, and constricted or flat, eggshell or terminal. We have now experimented and we found that these all are the dominant characters and these all are the recessive character. Then he did his experiments of hybridization because the flower's structure is also very easy and very easily the anthers can be removed or emasculated and the pollen are it's easy to transfer from the male part to the female part. So he used true breeding pea plant varieties. So the word is true breeding plant varieties. True breeding plant varieties means if you grow them again and again and collect the seed, they will be true breeding or they will give the same type of trait that was required in the experiment. So these varieties are used because he was going to study the exact transfer of or hybridization experiments in his further studies. So his studies are also called as inheritance of one gene or monohybrid cross, and he also studied the inheritance of other also, but in this part we'll go through the inheritance of one gene. Now you see he used two plants of one contrasting trait, that is height of a plant either tall or dwarf. So he crossed two parental plant, the tall and the dwarf. By crossing he obtained in the first generation or F1 generation, that is called as filial generation, all the plants they were tall. So there was no sign of dwarf plant in the F1 generation. So there was no sign of dwarf plant. So it can be thought that the dwarf is totally removed. But what he did now, he crossed the same plant or he did self-fertilization or selfing. After selfing, he observed in the F2 generation or filial 2 generation, 3 plants or the ratio as 3 plants they were tall and were dwarf, or the 3 part of all the plants that were developed in F2 generation they were tall and the one part expressed the dwarfness. So it was clear that in the F1 generation the character or the trait of dwarf was not completely dominant or it was not expressed, but the genes or the trait was there in the plant and it expressed in the F2 generation. So the same can be easily understood by this. This is there. Now if you see this, the tall can be written as capital T because these are, before going to this we can go through what is actual position of gene. Now you can say these are the two chromosomes. Okay, these chromosomes, on these chromosomes the genes are located. So the genes of the same trait are on one is on the one chromosome, another is on the another chromosome. These are homologous chromosome. So the contrasting trait of a same gene, we call them as alleles. Okay, these are alleles, and the position on which these are located, these are called as loci. So we have tallness gene, one capital T here, another small t here. The capital T denotes the dominant condition and small t denotes the recessive condition. The genes are located at different places. Here we see capital B means the genes are homozygous. Homozygous means they have same type of dominance, or they show dominant character. There is no hybrid form. Then in the C type of gene or allele, we will see one as a dominant, another as a recessive, another gene recessive. So this condition we call it heterozygous. And in the last one, that is D, this is small d and this small d, means this is a recessive gene and this is a recessive gene. So we call it as homozygous recessive. Homozygous because same type of genes are there, and heterozygous because different genes are there on the allele, and dominant because the dominant homozygous were there, and recessive because the recessive genes are there. So this is also one example, or the key words that you should know. Loci, it is a actual address of the gene. Allele, or they are taken from allelomorphs, alternative form of gene. It can be T, small t, A, small a. Homozygous, same locus, and they are same types, capital A or small a, small a. Heterozygous, dissimilar. Homozygous dominant, both should be dominant, both alleles of a gene at a locus similar and dominant. Both alleles of a gene, they are recessive. So on the basis of this, he started monohybrid cross. It is a cross between individual of the same species in which inheritance of contrasting pair of single trait is considered. So we'll go through only a single trait first, and then we'll see the dihybrid cross. So this way the trait or the monohybrid cross was done. The tall can be written as capital T. So what do we call this? We call this homozygous dominant. What do we call this? Small t, heterozygous dominant. So this is a genotype of this plant, and this is a phenotype of this plant. Genotype, the characteristic of genes, and phenotype, what it express. When they were crossed, we have F1 generation, and we got all the character as tall plant. But when we crossed, we see this T and this T. Now we have a hybrid, capital T, small t. When F2 generation was obtained by selfing, so we obtain the phenotypic ratio, that is the expressed ratio or expressed character as 3 tall and 1 dwarf, and the genotype character as 1 pure homozygous dominant, 2 heterozygous dominant, and 1 homozygous recessive. So by this way we can do the experiments, or we can write other by taking other character, contrasting character like flower color, like round seeds and wrinkle seed, like eggshell and terminal can also be taken. Like if you are going to take eggshell and terminal, we can write capital A and small a. If we are taking red and yellow, red R and small r. We cannot take the other letter because it will be more confusing to obtain the result. So on the basis of this, laws were devised, and these laws are first law of dominance and the second law of, that is, law of segregation. What is the first law? Suggests characters are controlled by discrete units called as factors. So he didn't call as genes, but he called as factors. These factors, they occur in pairs. You see here, these are the factors and they are occurring in pairs. In a dissimilar pair of factor, one member of pair dominates in a dissimilar. These are dissimilar. One is dominant and another is recessive. Only one character is expressing and other is recessive. The character that express is a dominant character. The second law, it states that the two alleles of a gene remain separate and do not contaminate each other in F1 generation. Okay, this is F1 generation. So this is 1, this is 1. So they do not contaminate. Contaminate means they do not show a different type of character. Either one is expressed or the second one. So here only one is expressing, that is capital T, and that's why the plants are tall. At the time of gamete formation, two alleles separate and parts into different gametes. So it parts into a different gamete at the time of selfing. We can write the law in this way also. The two alleles received from each parent segregate independently in gamete formation so that each gamete receive one or other with equal probability. So only one gene will enter to the to a gamete. So after doing this, the obtaining phenotypic and genotypic ratio, we also see some test cross. So what is this test cross? If we want to know whether the plant that is produced is a heterozygous or homozygous, heterozygous or homozygous, this can be determined with the help of a cross that is called test cross. So in a typical test cross, an organism showing a dominant phenotype whose genotype is to be determined is crossed with the recessive parent. The word here is recessive parent. Whatever the plant we are taking, if we want to know whether the plant is heterozygous or a homozygous dominant, then we should undergo a cross. We should do a cross between the recessive parent. This is a one example that is showing how the cross is made. Now the two situations are possible. Okay, this is situation number 1. This whole is situation number 1. So what happens if we are crossing a we want to know what is the genotype of this plant. So we are crossing homozygous recessive with a plant. Okay, and if all of them in the Punnett square, this square is called as Punnett square, and these all are the genes. The Punnett square, we found that all the plants, they were showing same character, means there is no ratio of violet or white, all are violet. So this means that the plant that we were determining its heterozygosity or its zygosity, we found that it is a homozygous dominant. This is the case number 2. If the plants that are resulting and they are in the ratio of 1:1, or 50% of them they are violet and 50% are white, then it means that the plant that we want to find out a zygosity, the plant is heterozygous in condition. That's why the results are obtained like this. So if the same type of character is shown, then we call it as a homozygous dominant, and if there is a ratio of 1:1 or 50-50%, then we call that character as a heterozygous character. So by the test cross, and the basic concept is this: it is homozygous recessive. It has to be crossed with homozygous recessive. Okay, this is case 1 and this is case 2. This is also the same experiment is demonstrated in the different way. You can see how it is expressing. So case 1 and case 2. If all are violet, then it is a homozygous dominant. If 50-50% are available, then the case is